Hello everyone, welcome to Get My Class. So let's begin the question here. The question says the technique of inducing variations in certain base sequences of genes by using chemicals or radiations is used in option A, selective breeding, option B, hybridization, option C, mutation breeding, option D, gene therapy. Now, let us understand the question. It says the technique of inducing variation in certain base sequence of gene by using chemicals or radiation. Okay, so where is it used? Okay, which among these will have this usage of this technique? That is the question. Now, first of all, we have to understand about the question, the key terminologies, whatever have been mentioned here. Inducing variation. What do you mean by inducing variation? Inducing variation means we are bringing about variation in the genes. That is nothing but mutation. We are bringing about mutation. And what is causing mutation? The chemicals used here or the radiations, they are the one which are bringing about the mutation. Hence, these are called as mutagens. Mutagens, because these can induce mutation. Now, so let us see. So where can we see the application of this particular technique to bring about variation? We want to change the general normal makeup for some, to create some variation, to produce some difference in the characteristics. So let us see. Option A says selective breeding. So selective breeding here, they don't do mutation. They actually select, in case of selective breeding, here, what do? They select the high vigor varieties, high or I can say, superior varieties, high or superior varieties, and further they will breed them. That is what is called as selective breeding. Okay. Now further we have hybridization. What do you mean by hybridization? Hybridization is also the same, like two different varieties will be taken or two different plants will be taken and they will be bred together and further it will produce a new variety. That is what is hybridization. So what is hybridization? Hybridization where two different plants having some different desirable characteristics, they will be bred with each other and that will give rise to hybrid variety. So mainly they'll be from different breeds, then only their viability will be really high. So that is how it is done. But again, here no specific mutation is done. Here also no specific mutation. Here mutation breeding, yes, this technique has been utilized in mutation breeding so as to induce mutation or so induce variation. Okay, so that is what I told you. So in case of mutation breeding, what do? They bring about mutation in the plant and further they will multiply the plant. So this is what is called as mutation breeding, where they induce mutation. Where do they induce? Mutation in the specific sequence, wherever they, based on the desired gene. Okay, here not all the genes are not affected, only the target gene will be affected. Okay, so this is the correct option. That is the correct option. Option D is gene therapy. Gene therapy is used to change the, you can say, whatever is varied already, that we are going to correct it. We are not bringing about variation. We are actually correcting the variation. Okay. And hence it is ruled out. Here we are not bringing about the mutation. Like we are not bringing about the mutation. Whatever mutation is there, that we are going to correct it, bringing back to the normal. Okay. That is what is gene therapy. So the correct option is what? The correct option is option C. That is mutation breeding. I hope you understood the explanation. Thank you, everyone.